Thank you very much, Andreas. I'm very, very happy to be here to share a different perspective that I hope will be of value to the audience. So today I would like to share two elements of what we are thinking about in terms of biomarkers. The first relates to the problem of how do you figure out what back to normal is unless you know what normal is in the first place. This really relates to some of the major challenges that we see in the traumatic brain injury space. So we heard this morning that TBI is, of course, a tremendous unmet medical need with many millions of injuries occurring worldwide every single year. And they encompass a number of different types of brain injury from a number of different causes. And the clinical heterogeneity of this disorder is a big challenge. So here is an example of some data from the CDC that illustrates different age groups have different susceptibilities to different causes of TBI. One thing I would like to point out is that very young children and older adults are believed to be at the extremes of risk for standard TBIs, from falls, motor vehicle accidents. We hear a lot and study a lot of brain injury in athletes, as well as in the military population. But there are valid questions as to whether or to what extent the data attained from these populations may be extendable to other populations. Also, I would point out that we know very little about the role of the aged brain in responding to traumatic brain injury. We know that when seniors fall and even have a mild brain injury, they can have a fairly rapid loss of quality of life. So one of the major challenges, of course, within the brain injury space is how to collect the premorbid data. Many studies are using control athletes, for example, and hoping that pre-season or unconcussed athletes are a reasonable proxy for people with no exposure to brain injury. But if you're studying an athletic population in the first place, especially as we saw this morning, 11 and 12-year-old boys learning how to play tackle football right from the get-go, it's not guaranteed that they're exposure-free. So how do we figure out what normal is? So we have, in Canada, a couple of large cohorts that might be of value, and I'd like to just introduce these cohorts to you. One of which, the program was really started by Khosrow Adeli, who is a clinical chemist working out of sick kids in Toronto. He is interested mainly in the pediatric space, interested in diabetes and obesity. But what he started was a large pediatric reference interval database program. So right now we have about 9,000 samples are available from children aged birth all the way up to age 19. And they've been characterized for a wealth of standard clinical chemistry biomarkers as it is now. So I'd just like to share what we sort of can do with this. So for example, one of the analytes that we have good data on is testosterone. And we have total, and we also have bioavailable testosterone. Now, in the brain injury space, if people are studying young athletes, this might become quite important. For example, often studies are simply stratified only on the basis of age, but you can see here that there is a very big range in the developmental maturity of boys at that age. Same goes for girls and their reproductive maturation. So we have all of these data. So these covariates could help be used to maybe interpret the data that are coming out of these sorts of studies. Now, it's important to realize that a biomarker concentration is not necessarily has the same level or pattern in age and sex. So I'll just churn through some other examples of how important a reference interval database really is. So for example, cortisol shows a very interesting wide range of normal values in the first early years of life. It tightens up and then it seems to start to spread out again as kids develop. whereas creatinine is a much tighter reference interval, on and on. So these samples are available in forms of collaboration for people who are identifying new biomarkers. Dr. Adeli has now put forward about 100 different FDA assays for the pediatric space into development, mainly through partnering with Roche and Abbott. And there is actually an app now that a physician can use to dial in a number and get the appropriate, what does that number mean? There's two other Canadian cohorts I'd like to introduce. One is the Canadian Health Measures Survey, and with the success of CALIPER, Statistics Canada, who runs this cohort, has been approaching the group to be able to use do the same sorts of things. So here, it's a nationally represented survey for the whole lifespan, and it spans age 3 to 79 years, and there are several cycles now that have been obtained. So if we take a look at some of these data, let's just go back. So this is an example of, again, the same types of reference interval standardizations that can be used. So it really helps. Like these lines here are the appropriate reference interval for a particular person in the population. So for example, a 37-year-old female, this is the reference interval here, but if it's a 17-year-old female, it's a very different interpretation of what is normal and what is out of range. And so each of these circles here in this database represent a conglomerate of 8 individuals. There are many thousands of individuals that are available. So essentially, we can match any patient with the appropriate age and sex, is really the bottom line. Okay, the final one is the Canadian Longitudinal Study on Aging. These samples have not yet been released for research, so we are actively seeking to develop partnerships to explore this. In this study, 50,000 Canadians are being sampled every 4 years between the ages of 45 and 85 years, and they're sampled at 3-year intervals. So far, there are about 30,000 of these samples available that contain biofluids. What's lovely about this study is that it will be longitudinal, and one can then track the progression of individuals over time at this age range. So these cohorts together provide an enormous statistical power to help the biomarker community develop and deploy clinical utility in the biomarkers that you're discovering. So I'm happy to talk about potential partnerships and collaboration for that. So that's on one end of things. Now, as a basic scientist, I'm also very interested in mechanisms and what we can actually do to experimentally understand how markers of the brain end up in the blood, for example. So here's Betsa Zlokovich's cast of the brain. So by his estimates, there are 400 miles of blood vessels within the human brain, which is a lot. And my laboratory is very interested in how metabolic factors, cardiovascular risk factors, affect dementia risk. And one factor that we are particularly interested in is the role of high-density lipoproteins, HDL, which you might know about as the good cholesterol because it helps prevent heart disease and heart attacks. It does this because it has several very potent vasoprotective functions at the blood vessel, which thus far have been studied really only in the large peripheral vessels. There is nobody, to my knowledge, that has taken this, as far as we have, into the brain. But there's a lot of epidemiological association between HDL levels and cognitive performance, AD risk, but the mechanism of how this occurs is not really understood. Then there's good preclinical model data as well supporting this. So we're very interested in this interface of the cerebral vasculature as reporting and integrating metabolic health with brain health. We know that conditions like type 2 diabetes, for example, are dementia risk factors, and we believe that this happens because the ability for the cerebral vasculature to maintain integrity of the blood-brain barrier and repair itself is diminished. So we're interested in developing ways in which we can actually study this question. in a method that does not involve animal models. So from our understanding of lipoprotein metabolism in particular, rodents and humans are very, very different. And those of you who are who study cardiovascular disease might know that you really have to subject mice to genetic manipulations to even to get them to get atherosclerotic plaques. So we believe that the neurological community is probably opaque to a lot of factors that affect brain health because we're relying on mouse models that do not report on some of these susceptibility factors. So what do people do instead? Well, they can grow endothelial cells in a static dish or in a transwell, and these are really widely used for blood-brain barrier models, but they're also very limited. They don't replicate the physiology of the vessel, they're short-term, and there's a lot of components that are missing. So our solution to this involves tissue engineering approaches. So I'll describe two of them that we have ongoing in the laboratory. So for example, we can take primary human cells and grow them in a scaffold-directed system in a bioreactor. So you end up with basically a vessel equivalent. And in this configuration, what I'll show you some data is on what would resemble a peripheral vessel. So it has smooth muscle cell layer and an endothelial cell layer. And then there's media that's pumped through this bioreactor like the bloodstream. The vessels are stable for culture 6-10 weeks at least, as far as we've really analyzed them. We can do a lot of really neat things with them. So this is what the tissue architecture looks like. We see a really nice tissue formation. It has the appropriate matrix, collagen, laminin, smooth muscle cell actin, and you probably cannot see it, but there's a monolayer of endothelial cells here, and they form a nice barrier to Evans blue. So we have quite a nice functional vessel. Now, we, because we're interested in Alzheimer's disease and inflammation, how inflammation at the vessel might impair brain health, we're doing all sorts of studies with this kind of system. So here, for example, we are adding A-beta to the exterior of the system, and we're looking at how A-beta added on the outside will trigger inflammation on the lumen by then flowing THP1 monocytes through the lumen, and you can see they stick to the endothelial cells in a dose-dependent way when A-beta 40 or 42 is added. In addition, what's really interesting is if we co-inject A-beta along with human ApoE on the brain side, or the exterior side, and we flow high HDL prepared from healthy human donors on the inside, we actually pull across the A-beta. So we believe that this is a combination of the HDL acting on one side and the ApoE acting on the other side to facilitate A-beta transport. This is just an example. We can measure transport in either direction, and we can inject whatever we want into this vessel configuration. So that's all very interesting, but it's not yet a cerebral vessel. So we've now enabled 3 cell types to grow happily together, and we're adding astrocytes to the exterior of the vessel. You can see the GFAP astrocyte layer here, and what look like end feet, which we're now confirming by electron microscopy on the exterior of the vessel. And what's interesting is when the astrocytes are cultured on the outside of the vessel, the endothelial cells then convert to a brain endothelial phenotype by turning on expression of blood-brain barrier-specific transporters. So now we have a nice tripartite system that resembles the natural human vessel. The endothelial cells are functional with respect to their nitric oxide production, and the astrocytes are phenomenally happy in this system in terms of the amount of apolipoprotein E that they will secrete. So now these little blue dots, these are the astrocytes. We really do have a brain side and a blood side now, and we can do the same sorts of studies. And we're interested, we have a handful of ApoE modulators that we're studying now in the system. But of course, there are many things, so this is just to show you that the difference in amount of A-beta that gets across when the astrocytes are there is much lower, it's faster, and so you end up with less accumulation of A beta into the actual tissue than if the astrocytes were not there. So that's a combination of the whatever astrocyte mediator is there, whether it's ApoE or something else, as well as the high-density lipoprotein on the inside that's really helping the system. So these sorts of three-dimensional dynamic physiological models that are entirely based on human cells, I believe, have a lot of utility in the future. We are able to do drug import studies for delivery of drugs into the CNS across the human BBB. And we can also look at what new biomarkers might be expressed and churned out from the blood side, depending on whatever you add on the brain side. So this is a very interesting space to understand mechanisms and identification of new biomarkers. We are able to help with drug development if there's cerebral vascular questions. For example, we know that one of the challenges with A beta immunotherapy is the development of ARIA, the vasogenic edema, in a certain select group of patients. So we might be able to help study that mechanism using such a human-based model. These are just sort of vignettes of what we're able to do in the laboratory, in the basic research and discovery arm of what my program does. And we're happy to work with anybody and showing them how to set this up. So thanks to the people in my group, the funding agencies, and thank you.
Amazing vignettes. Those are two gold mines of opportunity.
Thank you.
The first, just such critically important stuff for biomarker development. I have a question about your artificial vessels and the difference between A beta 42 and A beta 40. You didn't see any, it seems, in terms of the ability to cross. And yet, that seems to be a big part of the focus of new drug development, of trying to figure out whether monomers or oligomers are going to be the most toxic. Do you have a sense of why you saw the same thing in both?
So it's a fantastic question. So the parenchymal plaques are predominantly 42, but the cerebral accumulations are primarily 40. So there seems to be some difference in how the species the rate of transport across. We certainly know that there are subtle differences in the amount of endothelial activation induced by the species, but not yet the structure. So we're trying to do more defined studies. All of the data that I showed today were monomeric A beta. So we're now beginning studies where we add predefined oligos and fibers of the various, and then we can do mix and match sorts of things. So we're just at the cusp of being able to do these experiments, but I do believe that they're very, very important for understanding of A beta egress.
The normative value, this is obviously a great resource. So we don't have any data for large molecules yet, as I call it. So cortisol, so the hormones, electrolytes is what the current status of analysis is. Mm-hmm. Do we know anything about the history of TBI? Is this documented from a clinical perspective in these subjects?
Certainly, severe TBI would have been excluded. These are community samples, and so it was never designed to be a TBI study whatsoever. But what I would say is within the CALIPER, the pediatric cohort, these are active sports-playing kids. What sports they play and how much TBI exposure they might have had was never part of the study. But the protocol was designed in a way that we could go back and identify if there's any biomarkers that start to separate from other groups to be able to go back and do a sub-study. But in a way, this is entirely unbiased and does give you what the typical, at least kid in Canada, hockey-playing kid would do.